Thank you,
Thank
sir.
you
It's a honor
very much
to
giving
be there.
us the time, your valuable time, and joining with us. Thank you, sir. Yeah, please go ahead. Please take the chair.
Thank you, sir.
Thank you, sir. And thank you so much, Professor Suresh, sir. And dear delegates, sir is a research fellow of Durban University of Technology, South Africa. And in this today's session, sir is going to have a talk on plant-derived silver nanoparticles, recent advances, and transdisciplinary research perspectives. Sir, the team of SHCP and Sri Venkateshwara University welcomes you for this 2 days international workshop on recent advances and developments in transdisciplinary research. Welcome you once again, sir, and you can take over a session, sir.
Thank you, ma'am. Thank you, Dr. Surikitra, ma'am. And very good afternoon to all the delegates who are attending this international workshop. Myself, I am Dr. Suresh Naidu, working as research fellow in Durban University of Technology in the Institute for Water and Wastewater Technology. This is one of the main institute in South Africa, which focus on water research, algal research, and it has a multidisciplinary approach and different groups work in this organization with the different collaborations. And I would like to firstly thank for giving me the opportunity, the management, organizers, and especially Venkateshwara University Professor Narasimha Rao sir, who has invited me to share the some of our findings here on this good platform where any scientist will look forward to share. And I myself have been told to start the session and finish off by 1.5 hours, within 1.5 hours. So I have planned to complete as early as possible so that there is no time glitch and so that others can take the lead soon after my session is complete. I would like to thank once again, before starting, the management committee, organizers, and all delegates who are working hard to make it a successful event. So I have made my presentation in keeping students and young researchers and everyone who is interested in nanoparticles research and why it is very important to take up this one as one of the research area for further research and how we can achieve this. That is the main target. And before starting my session, I would like to share the video of IWWT Institute. It is a 6 minutes video where our director, everyone has a glimpse of DUT and what we do, the facilities here, so that you get an idea and we will share our knowledge as we disseminate the knowledge on this platform. Important international workshop. So I will start sharing my screen with the start of video so that after video, my presentation will start automatically. Thank you. This is my key topic, plant-derived silver nanoparticles, recent advances, and transdisciplinary research perspectives. Sorry.
Sir, excuse me, sir. We can't get the audio.
Yes, ma'am. Sorry.
Sir, it's not, audio is not there, sir.
From my side, it is 100% audio, ma'am. But my audio is visible, audible, ma'am?
Yes, sir. Yes, sir. Yes, sir. Kindly share the video along with audio, sir. In share screen option, kindly share the video along with audio, sir.
Sir, excuse me, sir.
Yes, ma'am.
Sir, we couldn't get the audio. Sir, while sharing the screen, share sound one tick mark we need to do, sir. Then only audio will be shared through the screen. Can you just do that, sir? From Zoom, when you are sharing the screen, share sound like one option will be there, sir. If you enable that only, sound will be shared, sir.
Sorry for the inconvenience, because I didn't want to stop that video, so that already we had hiccups with the technical. But can you hear me now, audio, ma'am?
Yes, sir.
Oh, sorry for the interruption. I think we can play at the end if the technical glitch is done. I don't know, but because from my side, audio, video, everything is perfect and it's going through, and I can hear the volume also. I don't know where it went wrong, but let me start my presentation, ma'am, so that the purpose will be served for the joining the session, ma'am.
Yes,
can
sir.
I take the lead, ma'am? Can I take the lead?
Sir, kindly share the PPT in presentation mode, sir. Slideshow. Click on slideshow, sir.
Slideshow.
The same one, sir. Slideshow icon. Yeah, click on that, sir. Give a double click.
Is it visible, ma'am?
No, sir.
Now?
Now it is visible, sir. Yeah, click on slideshow. Yes, perfect, sir. You can continue.
Thank you, ma'am. Thank you very much, ma'am. Dear delegates, students, staff, research scholars, today my topic presentation will be plant-derived silver nanoparticles, recent advances and progress in the transdisciplinary research perspectives in today's topic. I have planned this session focusing on students and research scholars who want to take up this area of research for further studies, and students will benefit to take up this aspect of research, because the end of research is to take the product to the community and publishing. So these two aspects are very important no matter where you do research. So communicating and delivering the product is a main important goal. Now, yesterday, before coming to final conclusion of my presentation, I just Googled how many articles are published on with the broad term silver nanoparticles. You see, if you look at this one, the first screenshot, in 0.08 second, 1.98 million results are there with the search word silver nanoparticles. That is the volume of knowledge body which is accumulating on this research area. And when you look at, if you narrow more, silver nanoparticles green synthesis, you are going to get 976,000 in 0.14 seconds. That is knowledge which is added. And if you go very specifically into the system, because nanoparticles can be generated from different biological sources. When I Googled for plants, it is giving 408,000 in 0.9 seconds. So this is the knowledge which is added on Google Scholar database. So like this, we have several databases, and each database will be collecting the related information, and it will be showing what is the latest happening in this related field. And now coming to this one also, if you see this particular article is cited 449 times. And if you see this article, it is cited 4,424 times. So this article was published in 2009, while this is published in 2013. Why I'm saying this one is, if you look at literature for the available of knowledge body on the silver nanoparticles, there is a report that silver nanoparticles from the last decade has picked up a more, and especially during the year 2012 to 14 and up to the 16, there was maximum number of articles published and available for readers. And gradually now, with the focus area and the delivery of the products, the articles are also coming less. So this is the indication what I wanted to give, but still there is very good scope for this area of research if you take up as a career. Coming to my presentation, I will introduce to what are silver nanoparticles, how these are isolated selectively, how they are characterized, and how a transdisciplinary approach can be used for getting the results. So for new student or one who is not aware of the nanoparticles, we can check lot of literature is there. Silver nanoparticles are class of nanomaterials. Sorry, 1 minute. I am unable to. Silver nanoparticles are class of nanomaterials with unique optical, electrical, and biological properties. So this compound is very much used for synthesis from biological sources, as it finds very important applications and with unique properties, optical and electrical. And we have to note that the silver use is listed in our culture from long dates. In the sense, back when we go, silver is applied for many purposes. And even in medicines and everything, you can see the silver application because it is having very good biological properties. What properties are exploited, we will discuss in course of time. And AGNPs have been shown to have wide range of applications, including medicine, food packaging, and water purification. These are some of the areas our center also focuses, but it has its own application. And in recent years, there has been growing interest in the use of plant-derived AGNPs because of biocompatibility, biodegradability, and low toxicity. Coming to our group, main focuses is on biomedical applications. So because I'm affiliated with IWWT and Health Science Department, we focus on necessity-based approach. And our group especially focuses on isolation, characterization, and assay for biological activities. And antimicrobial activity of nanoparticles, we exploit more, especially with silver and gold nanoparticles. And we do extensive study of cytotoxic and immunomodulation for synthesized nanoparticles. We do scan for antibiofilm activities. And some of the, where I'm working with medical school, where they check HIV integrase inhibitory activity of nanoparticles also in collaboration with the University of KwaZulu-Natal. And we do check anti-cancer properties with one of our collaborators in UKZN in Department of Biochemistry. And coming to the scenario of silver nanoparticles application, when you speak of the commercialization scale of nanoparticles, this figure is with respect to South Africa and to the rest of the world. When we look at the stages of research innovation, you will be having first generation, second generation, and third generation approach. So first generation, if you see, you will be having idea, you will be having development, and you will be having commercialization and manufacture process also associated with first generation. And second generation is covering idea and development of product. Third generation is nothing but there will be concept of idea, but it has to be materialized till it reaches to the commercialization and manufacturer stage. And in some of the South African research centers also, we fall in second category. So idea, research, and development process is there. And some of nanoparticles are also tuned for commercial application in CSR Center, Pretoria. So they check a lot of research applications for silver nanoparticles applications as such. And we have facility in Pretoria also, where a dedicated nano research center for excellences. Just for a common person to understand what is this scale of nano, what we are talking about in this whole lecture is, if you look at nano universe, it falls from 10 to the power of minus 7 meters to the 10 to the power of minus 10 meters. And especially, this is the nano universe where the particles which we synthesize also fall in this category. And to just give what is the dimension of other aspects, you see virus measures 10 to the power of minus 7 meters. And if you look at small ant, it is at 10 to the power of minus 2 meters. And these are some of the comparisons which are given for making a reader easy understanding. And this picture is taken from a source from ASU Eju. And why I'm giving this picture is just to get an idea. The particles which are synthesized fall in this range. They are not normally visible to the common eye. And they are to be examined with ultra, in the sense, sophisticated instruments for characterization purposes. Now, when you look at the official definition of the US National Nanotechnology Initiative, nanotechnology involves research and technology development at the atomic, molecular, and macromolecular levels in the length scale of approximately 1 to 10 nanometer range. So this is the range of nanoparticles we are discussing. And definitions are varying across the world depending on national strengths. If we look at China, Japan, and Korea, they emphasize the focus on materials, especially nanoscale materials and electronics, while researchers in Africa and Latin America often emphasize these nanoparticles materials in the context of medicine and environmental science. Means they are much focused on these aspects. While the Royal Society of UK makes the distinction between nanoscience and nanotechnology, where nanoscience includes study and manipulation of nanoparticles, while nanotechnology is nothing but design, characterization, and production of structures, devices, and systems at the nanoscale. So why I'm giving this statement in the sense, a reader should know what is nanoscience and what is nanotechnology, and what is the dimension of particles which we are discussing, and in which range we are going to deal with them. There are many significant advances and advantages also for plant-derived nanoparticles in recent years because of new methods that have been developed for the production of silver nanoparticles. Silver nanoparticles, in short, are referred as AGNPs from variety of plant sources, which coming from leaves, stems, flowers, and fruits. They are also called alternatively as biofactories in literature term. And these methods have resulted in the production of AGNPs with high purity and narrow size distribution. If you look at this picture, and this gives the various and the diverse applications of nanoparticles where they find potential applications. And the plant sources may come from leaves, it may come from fruit, or it may come from peel of banana, or you can focus on flower, or you can focus on roots, especially if it is a medicinal plant, you can extract. And what happens is from these plant tissues, the various biomolecules like proteins, amino acids, and polyphenols, terpenoids, organic acids, they act in the reduction of the salt, that is AgNO3, and you'll be having the applications. And some of the nanoparticles which are synthesized can find application in nanosensors to detect biomolecules, and they are exploited in water treatment processes, especially in the secondary stage where water pollutants are used for detection. And you can find applications in cancer, and we can see magnetic field, that is nothing but we can see some of the silver nanoparticles are used for imaging purposes, and some of the nanoparticles have potential photothermal therapy applications, some have photoimaging applications, some find cosmetics and medical appliances applications, and majority they focus on antimicrobial applications. And you can have sources coming from yeast, fungi, bacteria as a biological source for reduction of Ag, that is nothing but silver, for biosynthesis of nanoparticles. This is one of the recent work in collaboration with Prof. Narasimha sir and Prof. Kishore Bhattini from Padmavati Mahila University. This paper is now accepted, where they exploited the biosynthesis of silver nanoparticles from different sources: leaf, stem, bark, and flower, by reducing silver nitrate. And we will do characterization of nanoparticles, and we will evaluate antibacterial, antifungal, antioxidants. That is nothing but biological activities. And this picture, especially coming from graphical extract from medicinal plants, Nilomarchia kadamba, which is a local plant coming from SVU area. This is the approach to understand in detail how we will be having the silver nanoparticles production and evaluation of biological activities. Now, coming to the methods which are available for the manufacture of silver nanoparticles, we have different approaches. You can have chemical synthesis, you can have physical methods, and you can have biological approach for the production of silver nanoparticles. Especially chemical reactions are familiar for adding functional groups or adding additives for specific activities. So this is the chemical reaction method, a simple graphical representation where it will be coated with materials, and you will be having synthesis like micellase, and you can check for biological activity or use in the sensors. And in physical methods, you can use laser ablation method, metal plus solvent exposure, you will get different types of nanoparticles. And since we are focusing on silver nanoparticles, I'm not going into the structural details of 1 dimension, 2 dimension, 3 dimension nanoparticles. We are focusing on silver nanoparticles. So I'll make it simple for silver nanoparticles approach only. And when you look at biological substance, that is nothing but, since we are focusing on plant-derived nanoparticles, the source of nanoparticles it may be leaf, it may be root, or it may be flower, or it may be seeds also. So you can use anything as a source, and with proper reaction conditions, you can have biological synthesis of nanoparticles, which can find applications upon its suitability for and its biological activity. So this is a nutshell of general method which different labs adopt depending on their design and synthesis and their focus of research. And especially I'll be taking through our lab also, where we focus on recalcitrant seeds. Especially these seeds are very viable and very easily available, and they are not much exploited. So we select medicinally important plants as biological source. So the first approach in this presentation is isolation, characterization, and assay for biological activity. As I said, we select recalcitrant seeds. Recalcitrant seeds are generally big in size, and the picture is avocado, and this is Avicenna marina, a mangrove plant, which is common across the coastals, and almonds are also recalcitrant seeds. And what are these recalcitrant seeds are? They are very short-lived, which cannot be dried to moisture content below 20 to 30% without injury and are unable to tolerate freezing. So they are termed as desiccation sensitive seeds. Some common examples of plants that produce recalcitrant seeds are generally avocado, cacao, coconut, jackfruit, lychee, mango, rubber, tea, and some horticultural trees which are exploited. Avocado is a major crop in South Africa. So we focus on this avocado, and Avicenna marina, since Durban is a coastal we focus on this medicinally very important mangrove plant. And you might be knowing that most of the research now are going for exploring marine areas, especially which are not explored, either from sponges or from marine source of plants. Now, coming to biosynthesis of silver nanoparticles, just I will explain the brief steps which are involved in the biological synthesis of nanoparticles. First, you have to identify the source for your experiments. So since we have selected, and I will be focusing on seed samples, which we normally do in our labs, will be collected and washed thrice with tap water and twice with distilled water to remove adhering salts and other associated contaminants. So the cleaning is must, and before you start any experiment with any unknown medicinally important plants is also that you have to take care of proper identification by taxonomist and submission its herbarium to the concerned department, and then you have to proceed with the this step. Or otherwise, sometimes you may end up with the alien plants which are not identified. And once you go to publication stage, they will ask all these details, whether it is identified or submitted to the concerned department or identified by experts. All these questions will start. So before start only, you have to identify with a taxonomist your target plant and confirm it. Then go through normal available literature, which is available for leaf. So you can use roots or you can use stems, any other source, and proceed with literature which is available, and you can modify the steps according to your need also. So standard step is you need to have a proper way before you start experiment. So we started with 10 grams of seeds. So it will be finely cut into small pieces and boiled with double sterilized distilled water for 5 minutes. This is a common procedure which we use silver nitrate at a concentration of 6 millimolar, and it will be incubated at room temperature in the formation of brownish yellow color, indicating the synthesis of silver nanoparticles. So this confirms after incubation with silver nitrate, your biological source, you will be having color change. So this color change will confirm that silver nanoparticles are formed. And after that, your 1 ml will be taken out and it will be scanned for UV visible spectrophotometer in the range 300 to 700 nanometer at 5 minutes time interval. At different times intervals also you can carry out if you are studying for optimization studies. And if you are not sure whether it has fully formed or not, leave it for overnight. 8 hours of incubation should assure you that there is complete reaction set completed. And once these particles are formed fully, you can do centrifugation at a specific time and take out your supernatant, wash it with distilled water and dry it, the nanoparticles which are formed, and you can carry out the further studies for your research. So this is a simple approach where we have published this paper using Avicenna marina seeds, which is a mangrove plant. And what happens during this process is the biological source which we use has a lot of biomolecules with different functional groups. And these are some of the class of biomolecules which are generally involved in the reduction process. When you treat Ag1O3 with biological materials, so the functional groups reduce silver to silver ions, and only very few I have listed with their chemical structures and with the functional groups. So proteins, phenolic acid, saponins, luteolins, amino acids like tryptophan, tyrosine, and ascorbic acid, and eugenol, sterol. These are all the compounds involved in the bioreduction of the silver nanoparticles. And when we focus on any biological process, and there will be several factors associated with the formation of nanoparticles. So when you process or when you optimize the step only, you will be having better yield at the end, or otherwise you may end up with the very low quantity of the nanoparticles, which cannot be further characterized by advanced instruments. So generally, if you start with 500 ml, you may get very less quantity of silver nanoparticles. If you start with 1 liter, something like that, you may end up with good quantity of silver nanoparticles, which can be characterized, and you can go for further studies, whatever you are required for biological assays. So this is a common path, which is where I have shown a plant extract mixed with silver nanoparticles, and there will be production of nanoparticles with low yield if you don't optimize the condition. So the next step will be optimization step, and there are several parameters for the biological synthesis of nanoparticles. Some of the factors which influence is incubation time, the mixing ratio, temperature, pH, and aeration. And if you study the details of methods, several teams will first optimize this process so that they have better yield. Once you have stable production after optimization of all these parameters, then it will be going for characterization step. And the nanoparticles, what you get, will have different shapes and different sizes. So remember that when you scan the nanoparticles, it should be less than 80, 90, or 50 nanometer. That is a preferred category of nanoparticles, so that you have some biological activity. And there are reports on the shape and size of nanoparticles which influence your biological activity also. For details, you have to read some concepts which have explained how the shape and size of nanoparticles influence the further research. That is nothing but biological assays. So you will be having square shaped, you will be having spherical shaped, triangular shaped, hexagonal, and rod shaped crystals, that is nanoparticles upon biological synthesis. And when we see this step, that is nothing but nanoparticle production, some groups or some literature say it is a top-down approach or it is a bottom-up approach. So in this figure, I have listed what is top-down approach and what is bottom-up approach, and different methods which are available when you look at the literature. And this is coming from one of our students from UKZN, who was honor student, where she has written for thesis, and this was generated. And it has gone for publication also into a specific book chapter, where we have listed all the widely used methods for synthesis of nanoparticles, especially physical methods like evaporation, condensation, laser, splattering, milling, explosion processed, and chemical methods. Some of them we have listed is wet, sol gel, vapor deposition, spray, pyrolysis, aerosol processes. And biological, especially, we have focused on plants, microbes, seaweeds, mushrooms, and cell-free extracts we have used for biological methods. And when we compare both the methods, you will be having limitations and advantages also. When you see limitations, if you look at physical and chemical methods, the physical methods demand high energy requirements and large working space and imperfections on nanoparticle surface. And when you look at chemical process, there will be involvement of use of toxic chemicals, and lot of aggregation is reported once nanoparticles are formed, and generation of toxic byproducts also. So this is the reason why now most of the research are shifted towards biological approach, that is nothing but green synthesis. The green synthesis approach is mainly advantage because of its non-toxic nature, eco-friendly, cost-effective, and biocompatibility of the produced nanoparticles. So this biocompatibility is very much advantageous when you evaluate in vivo studies on nanoparticles. Now, the second step in your research approach should be characterization of silver nanoparticles, which are synthesized by either chemical method or physical method or biological method. So I will take you through some of the major steps, in the sense, major techniques which are involved in the characterization of silver nanoparticles. So the first key instrumentation will be UV spectroscopy of silver nanoparticles, which will give you idea whether you have formation of silver nanoparticles or not. Though we can judge by physical observation the color change in the reactions, that is not sure. So we have to scan the nanoparticles which are formed in the aqueous solution. So after 10 minutes, or some reactions are so fast within 2-3 minutes. So once you add your biological source, there will be color change. But from our experience, it is better to leave overnight and take the readings so that there is full reaction and full reduction of the nanoparticles. So this is how a control, and this is after treatment Now, when you scan this UV spectroscopy for your reaction, that is biologically synthesized nanoparticle suspension, there is a process that is nothing but SPR, which is a technical term, so that the nanoparticles have specific absorption between 400 and 500. You can see the peak here, specific peak point, where it confirms the reduction and the formation of silver nanoparticles. This we have published. And if you have different sources also, you can scan separately. And this is one UV spectral scan where leaf, stem, bark, and flower were used, and you can couple the reactions. And you can see each one will be having their own specific peaks because depending on the starting material. If it is leaf, there will be slight variation, but the main activation will be between 400 and 500, especially at 430 nanometer, you will be having specific absorption because of surface plasmonance resonance effect. The nanoparticles start exciting and the electrons start showing the positive indication of this production. The other versatile technique to confirm the nanoparticles formation is scanning electron microscopy. So this is one of our approach with the marine plant. You can see some image of formation of silver nanoparticles, the method which I have explained. And you can see along with silver, other elements are also picked up in your process. So if you want only silver, you have to adopt a modification approach to purify the or to overcome these elementals. But this is sufficient to confirm that there is production of silver ion which is confirmed through scanning electron microscopy. So I'm not going into the instrumentation details, but I'm telling you what techniques are which can support your research, or if you take up the characterization of silver nanoparticles in simple strategy. Now, the second most widely used technology in the sense for characterization is transmission electron microscopy. If you do TEM of silver nanoparticles which are produced, you can see the triangle-shaped nanoparticles which we have got from Avicenna marina seed extracts. So you will be having different shapes, but you have to scan and you have to take notes of the nanoparticles which are found, and you can correlate the biological activity with the shape and size. So this fall at 50 nanometers, and this is at 100 nanometer resolution, the nanoparticles, how they appear from different conditions. So we have changed and we have scanned, but I have shown a collective picture to get the idea that how TEM pictures of silver nanoparticles look and how the shape of nanoparticles will be exactly when you do TEM. Another versatile technique which is commonly used is FTIR spectroscopy. What this technology helps is, by using FTIR spectroscopy, you will be having specific wavelength excitation and you will be having peaks. And there is a database where you can compare the peaks, what you got, and you can predict the functional groups which are associated with your compound. So this is how we will go back to once again the chemical structure of the compound. So if you see the peak values of specific what I have shown here, that is 3333 belongs to functional groups ammonia. So it is a conformation of aliphatic primary amine. So hydroxyl groups will be having 3305, which is responsible for carboxylic acid. So the database, will scan and it will give you a picture like this, and an expert who is a chemistry with a chemistry background can read your graph and give you the functional groups which are associated. And this is how finally the molecules and the shape, structure, and functional groups which are involved in the bioreduction process can be reported in your paper. So I have listed a few common functional groups which I have taken into consideration and are responsible, and which we have published in Bionanoscience Journal. Another technique which is very vital, and it will solve most of the problem. Most common complaint in labs is the aggregation of silver nanoparticles because of the charge. So zeta potential is an important and readily measurable indicator of the stability of colloidal dispersions. This is very important factor. And this picture explains what is the surface charge, what is the stun layer, and this is a slipping plane. And the potential, zeta potential, is a charge, and it gives indication of the stability. So if you have zeta potential after scanning your solution or suspension, or the silver nanoparticles which you have ready for characterization, if it displays 0 to 5, you are going to have a coagulation process or flocculation phenomenon. That is nothing but aggregation, self-aggregation, and which is not preferred in nanoparticles research. If you have 10 to 30 as a zeta potential, it is low, and it is maybe regarded as instability also. If you have 30 to 40 zeta potential, it shows moderate stability. If it is having 40 to 60, it expresses good stability. If it is above 61, you are going to have excellent stability in the product. So zeta potential is one of the key criteria which is adopted for checking its stability, especially in the colloidal dispersions. That is the in biological approach. And there is another technology which is commonly linked to X-ray, that is energy dispersive analysis, which is called as EDAX. This is a small instrument, but it gives very vital information. If you look at how it picks up the presence of ion, and the samples are coming from different sources as listed in the figure: leaf, stem, flower. You will having, I'm unable to see the screen here, but the peaks confirm the presence of Ag ion, and some other ions are also picked up in this data. So energy dispersive analysis, that is EDAX, will confirm the presence of silver ions in your sample. These are all required when you start working into the wet lab, especially if you start a research career in nanoparticles research. This is the very simplest nanoparticles research. And this table will list you the different techniques which I have discussed. And DLS I have not discussed, dynamic light scattering. And this also measures the size. And if you look at TEM, it gives the information regarding size and shape and preparation subject to artifacts. Some of, if you don't have expert, that is a dedicated expert, you may misinterpret the data. And there is another advanced technique which is called as AFM, that is atomic force microscopy, which also attributes to size and shape measurement, and limited by cantilever tip. This cantilever tip is the tip which is sold by many vendors with different sensitivity. So if you have the good model, then you are going to have very good data which is supported with AFM. And zeta potential, surface charge will be indicated, and it is mainly affected by capping agents and shape. And there is another measurement which is not so routine in the labs, Bronner-Emmett-Teller, which gives specific surface area and measures total area available for interaction. And disadvantage is it cannot predict the size distribution. So these are some of the information which I have gathered for student purpose, so that they can have an overview of different techniques, and the techniques which I have discussed previously will add value to the characterization steps which they have to adopt when they take up the nanoparticles research. And some of the common biological assays for therapeutic potential, all labs, and especially in our lab, we study both in vitro approach and in vivo assays. So in vitro toxicity assays are oxidative stress assays, DNA damage assays, which is commonly referred as COMET assays, cytokine production assays to see the immunomodulation approach. And we adopt cell line cytotoxicity assays also to study the anti-cancer properties. And some of the in vivo assays, which are done in collaboration to check effects on circulatory, respiratory, and central nervous systems, are adopted. And especially for wound healing properties, we have collaboration on dermal tissues studies, and we can do hematological parameters. And especially in the growth and development effect, we can adopt zebrafish development assay. And skin, porcine skin is adopted for different approaches, but for wound healing also, we approach this way. And these are the common rats. Suppose if you are studying anti-diabetic effect, we need to have specific strains meant for this kind of study. Because now nanoparticles after production, they do different biological assays to get the information where it is more active. So these are the common and widely applied techniques to get the information on your nanoparticles, which are synthesized by biological approach or chemical or physical. But I'm focusing on biological approach. And another major application of this nanoparticles approach is they have very good antimicrobial property. So these are some of the highly listed pathogens by WHO, where one can start focusing with specific strains, especially, and their antimicrobial effect. Common is Pseudomonas and Enterococcus faecium, Staphylococcus aureus, Heliobacter pylori, Campylobacter salmonella, and Neisseria gonorrhea. This species is most extensively studied in UK area, in the sense of especially those labs focus on this aspect. And coming to our lab, we have different strains in collaboration, and strains are available with specific collected from ATCC, so that we can focus on necessity, whether you want to do in consortium or whether you want to do individual. If you want to study single, you can take pathogens such as E. coli and study, which is a normal infecting agents, and study its properties. So like this, these are some of the red listed pathogens recommended by WHO, which we focus for our study also. And now, all studies, when we do any biological approach, we need to have a foundation why we have to approach and how we can approach. So the most established study aspect is the toxicity mechanisms of nanoparticles, which is mainly mediated by ROS, response oxygen pathway. So when silver particle or silver particles are exposed to your live cells or living organisms, there will be penetration and there will be incorporation in the mitochondrial damage they are involved, or they may be rupturing the electronic membrane and result in membrane damage, or the ROS response pathway can initiate to inflammation, cytotoxicity, apoptosis, protein denaturation. And if it enters into DNA, there is a probable DNA damage and genotoxicity, inflammation can be resulted. So these are all the pathways where silver nanoparticles are evaluated for their biological interference. And this is reported, well-documented steps. And now, coming to the theme, that is nothing but the first part was focusing on plant-derived silver nanoparticles, isolation, characterization. Now, let us see why we need to have transdisciplinary research approach. The research on plant-derived nanoparticles is in its early stage. So we need to have transdisciplinary approach, and there is a need because it brings experts from different fields such as chemistry, biology, medicine, and engineering. Because most of the applications we can see in nanosensors, which is a mixture where you are having a chemistry expert, where you are having a physicist expert, and engineering, and ultimately the application will be on medicines and or environmental scanning, pollutions. So this picture shows at nanoscale, you will be having intersection of physics, chemistry, and which can lead to, if you explore at this point, that is nanoscale level, you are going to have new markets, that is nanoparticles which are synthesized. So new markets can be generated by using the physics laws, biological principles, and chemical properties. So this is a point where transdisciplinary approach can be adopted. This is one area, since we are focusing on plant-derived silver nanoparticles, there is a separate discipline which is called as phytopathometry. This is measurement science in pathology. And this has direct broadcasting on measurement of disease, its infection, epidemiology, and forecast the prediction of future seasons are also predicted by the use of this discipline, where you can see physics is playing, sensing technologies are incorporated, artificial intelligence is incorporated, robotics are incorporated, where we are giving forecast regarding disease resistance, phenotyping, and particular epidemiology, which can give agronomy a science where you are predicting the loss also for future. Or you can alert agriculturists by this approach. So this is one of the recent development, which is very well known science in agriculture, phytopathometry, which incorporates different disciplines, including statistics, physics, and pathology experts, imaging and sensing technologies to broadcast particular disease, especially in the field of agriculture. So there is interrelation, pathology, measurement science, psychophysics, imaging, and sensor technology. In a nutshell, this picture depicts the transdisciplinary approach which we can adopt in the future. And this is already in practice in various universities. And now, what we can achieve in transdisciplinary approach. And this is a integrated innovation model, which is already in practice in Arizona University in US, where they have incubation centers, especially Western countries like US and China. They focus more on nano-related products. So you can see their nano products in markets. On the shelf also, you can see they have cleared the first, if you recall, that South Africa is second generation, but the first generation are the countries where they have idea, development, and commercial. So first world countries have this model of incubating where they have different responsibilities and they can achieve through different integration models. This is appreciated by the corporation, and this is good for society and environment. This can be achievable through engineering concepts, integrated, and the product will be value added to the consumer. So this is an integrated innovation model for responsible product development, which is adopted in Arizona University. And let us see how there will be societal development and impacts on the nano by the use of nano. If you see, the nano science is integrated in three sectors, that is academia, industry, and society. We fall in academia, and there is integration of industry and society. That is the outcome. So what an academia person can benefit by transdisciplinary research approach is you will be having research collaboration, which is multidisciplinary. So this is the future science. As a Dean, SVU, Research and Development has highlighted that the future of research is for collaboration. So we also look for good collaboration so that we have more benefits. And you will be having applications developed through multidisciplinary approach focused on restoration, environmental damage, which is already done. And you can bring awareness in the public regarding the understanding and the use of nanotechnology. So these are the major role where academicians play vital role. And if you look at the benefits by this transdisciplinary approach is hello. Thank you. There was I think someone has opened the mute option. When you look at industry, ultimately the benefit of the research innovation in nanoscience and nanotechnology will result in low manufacturing cost. And there will be improved productivity of any product, and there will be new industries coming across the scenario, so that you will be having enhanced product. And what happens to the society is you will be blessed with improved standard of living. There will be economic growth and capabilities of an individual or community. And you will be having a strategy for preventing, diagnosing, and treating diseases, and strengthen the national security by certain nanotechnology also. It is recorded. And this is how we are going to have the different societal development by the use of transdisciplinary approach in our basic research. And coming to nanoparticles application, I'm speaking of general approach, not only derived from plant. So if you see health and fitness sector, 53% is captured, electronics and computer 4%, food and beverage 8%, automotive industry is 9%, home and garden is 15%. So this is the market capture of nano-based products, especially silver, which has captured in the market. And it is a multi-billion-dollar industry in Western countries. Here in this table, I have listed some selective applications coming from silver nanoparticles synthesized from medicinal plants and common plants for their effective. If you see moringa, which is a common, widely used in Indian continent and everywhere. If you, there is a report for treating cervical cancer on lab scale. So there is a drug delivery approach by using silver nanoparticles, which are synthesized. And from Catharanthus roseus, they have reported anti-plasmodial activity using silver nanoparticles. And Philanthus neruri, they use silver nanoparticles. They have reported mosquitocidal properties against the dengue vector Aedes aegypti species. And from this medicinal plant, Epilomascus moscatus, I'm not basically taxonomist or botanist, so a little bit my spelling will be different, the pronunciation of the name. It shows wide antimicrobial activity against the selected pathogens. And Rhizophora, silver nanoparticles, you will be having nanoparticles which are reported to be very effective against the bone cancer. And Atropa, they have produced controlled autoantigen production, and which is involved in the information process. And there are many reports. If you look at the literature, the first thing widely used is antimicrobial properties. So antibacterial, antifungal, and silver-based drug delivery systems are exploited from plant sources. These are all commonly reported in the research body. Just, I would like to conclude the presentation. Plant-derived silver nanoparticles have a wide range of potential applications. Transdisciplinary research is needed to accelerate the development of these applications, and this is the need of the time. And the future is only transdisciplinary research approach, not only to this field, and it applies to several other fields also. This research has a potential to revolutionize the way we use nanoparticles in medicine, food packaging, and water purification. These are major areas where they find extensive application. Some of the challenges we should not neglect. One is toxicity. If you use in high concentrations of silver, there is a lot of discussion and debate on the use of silver nanoparticles. And the yield of the synthesized nanoparticles corresponding to the metal salt concentration has to be optimized, and the available biological resource remains to be elucidated. And the parameters that can overcome the problems of polydispersity is a challenge in biologically synthesized nanoparticles, which still require optimization through different biological systems. This is one challenge. And other challenge is issues relating to the biomedical applications of nanoparticles, including distribution profile, excretion, and clearance of nanoparticles in vivo trials need to be addressed. So these are the conclusions of the today's topic. Just I will take you through some of our selective publications from coming with our research collaboration. And this is advanced article, which is already accepted, and we are waiting for proper indexing. This paper, especially green synthesis, characterization, and biological activities of silver nanoparticles from Kadamba medicinal plant, is done in research collaboration with Prof. Narasimha Sir and Prof. Kishore Batini from Padmavati Mahila University, which is accepted, and it is due for full article length. And this was done with UKZN when I was a postdoc. We in this research, we have used different physicochemical optimization, evaluated antimicrobial and anti-cancer properties, especially silver nanoparticles, synthesized organ-specific from Bidens pilosa. So P. Govender is our supervisor in UKZN. And this is done with one of our collaboration, Dr. Subhendu, where we established and the research was the nanocomposite synthesis by hydrothermal method, which we have published in NJC. And this is a review article coming from one of our collaborators, Rajneesh Anupam from Gaur University, which is the Central University of Technology in Madhya Pradesh, where we have reported several organosulfur compounds coming from sulfur, garlic especially. And this is another paper which we have published where we have focused on synthesis from potato, iron oxide and manganese oxide nanoparticles, and evaluating their effect on the using tissue culture approach. And this is coming from our lab, where you can see Prof. J.K. Adam is my supervisor and line manager here. And this the concept which I have explained, Avicenna marina seed extract and its antibacterial potential, was published in Bio-Nanoscience coming from our lab. And this is also one of the area where we focus, and this was done with Prof. Rajshekhar Reddy sir, and Narasimha sir was our research mentor in this paper. And this is one of the paper which we have published in Current Trends in Biotechnology and Pharmacy, where engineering nanoparticles, hazards and risk assessment was reviewed. And this is another paper of gold nanoparticles, which was published in Transactions of the Royal Society of South Africa. And we have collaboration with University of KwaZulu-Natal, Rhodes University, and Hari Singh Gaur University, and we are working effortlessly to collaborate with Sri Venkateswara University at official level. But we have extensive research collaboration with Prof. Narasimha sir's lab, especially Department of Virology, and we have published a couple of papers and book chapters under his guidance. And Prof. Narasimha sir is my senior, and I did my PhD from Anandpur, SKU. And right now, we have a project which is running with the support of UK Newton Fund, which is focusing on antibiotic accelerator initiative. This was awarded to Prof. Rosemary Durrington, and I am representing from Durban University of Technology. And this is our institute picture. Just I would like to thank you everyone to be patient and listening the talk. You can discuss anything via email, and I'm available for any queries. And the duty is ranked in top 5 in South Africa and #1 University of Technology in South Africa. This is the latest appreciation given by Times Higher Education. I would like to thank and close the talk for today. And I know we had some glitch, but we overcome that. And if there are any questions, I will take the questions. And I would like to say thank you for the organizers and the committee for accepting my proposal to present here in this international workshop. And I think all the audience will get and take home message with new ideas which are presented in this venue. And we are looking forward for any queries. Thank you.
Namaste, sir. This is Dr. Neeraja.
Yes, ma'am.
I am from KVR Government College, Cluster University, Kurnool. And your presentation is excellent. It has given really a wonderful dimension to our research also. And I would like to invite you for, we are sending the project proposal for smart tribal farming. In that, I would like to include this one, the phytopathometry in agriculture. And this is the benefit of attending your session, sir. I would like to include this one. And I invite you for the collaboration if we get the project. And number 2 is, can you brief the work you're doing there with this nanoparticle in connection with the water pollution and all? Just and how you use this in, you have elaborated on how it is useful medicinally. And I'll be very happy how it is used for the pollution control. Just a brief note.
Yes, ma'am. Yes. Ma'am, actually, in our institute, IWWT, that is Institute for Water and Wastewater Technology, we have different groups. I am associated with microplastics and nanoparticles research, and we focus, and especially myself, we evaluate biological activities, and some of my colleagues are directly involved in wastewater treatment at different plant levels, and they are they will be the right person, but we have initiated FICO nanotechnology where algal sources will be exploited for waste treatment. Especially, we are focusing for dairy effluents and secondary stage treatment of water treatment. Secondary stage. And the right person will be my colleagues, because I have not initiated that work, but we work at biomedical applications, to be frank. But my email is there, which is now displayed, sureshk@DUT. You can drop me email, then I will put you in the right contact. So you can start exploring the research area, or you can ask me with your specific questions. I can send you the access what I have and how we can go ahead with your research work also.
Thank you so much. Thank you. Thanks a lot.
Thank you, ma'am.
Sir, thank you so very much for your wonderful presentation, sir. And we have one query in the Zoom chat box, sir. Can you read that query, sir? So the Ashwini madam is asking, can we apply this approach for fungi?
Yes, ma'am, definitely. Since the talk was more on plant derived, I have not shown where some of our collaboration is coming from Christ University, also from the Bangalore, where Prof. Atika and Dr. Vasanta Lakshmi are involved. They are exploiting, in the sense they have used fungal templates, synthesis and application, but with biological activities like antimicrobial. And I can put you in the connection, and definitely fungal also act as a very good source, because there are reports by the use of Candida species, they have synthesized the silver nanoparticles. And different organisms, first thing is we need to identify the source and what is your problem and how you can find the remediation. So by this, we can have a go-through approach for silver nanoparticle synthesis, ma'am. Yes, there are reports on fungal nanoparticles also, and we have published. And I just didn't show that one because it will be not aligning with the talk. So that's why I avoided. But we have publication with the fungal activity also.
Wonderful, sir. And you can also access all the messages in the chat box. And all those accomplishments are really a reflection of the intensity of your presentation, sir. Very nice to have you. And sir, we have another one more question from one of the delegates, sir. And the question is, can we use nanoparticle to bind with processed millet food to digest it completely? You can see the question in chat box. You can see.
From whom, ma'am? Because I'm scanning now.
Okay. So can I repeat the question, sir?
Ashwani, I have seen very information. And
Dr. Suresh, this is me.
Hello. Yes, sir.
Dr. Suresh, this is me. Actually, I asked this question regarding the fungal nanoparticle.
Yes, sir. You can go ahead, sir.
You already answered.
Okay. Thank you, sir.
Yeah.
Thank you, sir. And this is a query from Sharon Kauda, sir. Can we use nanoparticle to bind with processed millet food to digest it completely?
Yes, this is a little bit concerned because as I say, the nanoparticles, if you use in conjunction with processed millet food, it is not suggestible because there is concern of nanoparticles direct exposure to your body, and which is not suggested. That's why silver-based products are not in popular use because of this concern. And especially biomagnification or bioaccumulation is going to happen if you take it along with nanoparticles. That is not a suggested route. And Dr. Sharan Gaud, sir, is our, I don't know what to say. He is my friend. And I'm aware of Dr. Sharan Gaud, J. Patil, what I can say. Thank you very much, sir, for your question.
Thank you, sir. Thank you so very much. And we have another one more session speaker is waiting on the line. So very sorry, delegates, I'm moving forward. And now I request Mrs. B. Shivagami, madam, to propose oath of thanks for today's session.
Yes, ma'am.
A very good afternoon to all. I am Mrs. B. Shivagami, working as Associate Professor in the Department of Pharmaceutical Analysis, Seventh College of Pharmacy, Tirupati. And I take this opportunity to thank our beloved Principal Sir, Dr. M. Niranjan Babu Garu, and correspondent Sumalata Ma'am, and for their continuous support. And I thank all the dignitaries of SV University for their support. And I thank today's eminent speaker, Professor Suresh Babu Naidu, Krishna Sir, for delivering a wonderful session. Sir is a research fellow at Institute for Water and Wastewater Technology, Durban University of Technology, South Africa. Sir is a member of Royal Society of South Africa, the South African Society for Microbiology, the South African Society for Biochemistry and Molecular Biology. Sir received Faculty Researcher of the Year in Gold category Health Science 2022. Sir is a recipient of University Capacity Development Grant. Sir received Top University Publisher Award in 2017. Sir received Top University Publisher Award in 2016. Sir is a lead scientific researcher on 50+ scientific research papers. Sir is a reviewer for research proposals for funding agencies. Sir is a keynote speaker in presenting speeches at international conferences. Sir, thank you so much for joining with us for this 2 days international workshop, sir. Thank you so much.
Thank you very much, ma'am. It's an honor to join the community of Seven Hills, and it's a revisit to my native place. I am basically from Nellore, and my blood connection now sister stays in Vijayawada. Last year I visited SVU where there was an ICON conference in the campus, and I have collaboration with Prof. Narasimha sir. So constantly we'll be talking about the research, and I'll be taking this guidance, and is our well-wisher. And as far as right from lab stage, he is my senior guide. Thank you, ma'am. Thank you, Principal Sir, for the opportunity. Thank you for all the community of Seven Hills for giving this opportunity. Thank you, sir.
Yeah, I think Dr. Sucheta, madam, you can present a memo too.
Yes, sir. I request you to kindly do the honors to Suresh Naidu, sir.
Yeah, sure, sure. Yeah, good afternoon, everyone. I am honored to be here to felicitate our resource person, Professor Babu Naidu Krishna Garu, Durban University of Technology, South Africa, for his outstanding achievement and contribution to this field. Heartily congratulations to you, sir. Please accept this memento for token of respect. Thank you, one and all, for joining with us. Have a wonderful day.
Thank you, sir.
Thank you, sir.
Thank you very much, ma'am.
Namaste.
Thank you. I'm leaving the session, ma'am. Thank you.
Sure, sir. Thank you so much, sir. Thank you.
Thank you, ma'am. Thank you.
And delegates, moving into the session 3 of this today's 2-day international workshop. We have our eminent speaker with us today for the session. Sir is Dr. C. Ronald Darwin, sir, and currently, sir is a professor and head of Wales University, Tamil Nadu. Sir, welcome you for this international workshop, sir, and very good afternoon to you, sir. Sir, good afternoon to you.
Good afternoon, everyone. I would like to thank my most lovable friend, Dr. Sharavana Kumar, for initiating this opportunity. Am I audible?
Yes, sir.
Yeah, you are audible, sir.
Okay, fine. Thank you. It is a wonderful opportunity given to me. Can I start the presentation directly? Sure.
Just a minute, sir. We have a welcome address, and followed with that, we can start the presentation.
Fine. Thank you. Thank you.
Respected delegates, very good afternoon. And for this today's session, we have our resource person, Ronald Darwin, sir. And sir is going to have a talk on role of artificial intelligence in drug discovery. Sir, I welcome you on behalf of Seven Hills College of Pharmacy and Sri Venkateshwara University, sir. Thank you so much for joining. And I now request Dr. S. Divya to read the citations of Ronald, sir.
Thank you, ma'am. Good afternoon to respected principal, sir, today's speaker, and dear delegates. I'm Dr. S. Divya, Assistant Professor, Department of Pharmacy Practice, Seven Hills College of Pharmacy. And it's my privilege to introduce one of the speakers, Dr. C. Ronald Darwin, sir. Sir completed MPharm Pharmacology and PhD from the Tamil Nadu Dr. MGR Medical University, Gindi, Chennai. And formerly, sir worked in KK College of Pharmacy and in Mohammed Satak AJ College of Pharmacy, Chennai. And currently, sir is working as professor and head Department of Pharmacology, School of Pharmaceutical Sciences, Wales Institute of Science Technology, Chennai. Sir guided many PhD students, MPharm projects, and also many BPharm dissertations as well. And also supervised and guided 32 MD Siddha research projects as well. Sir is having 6 patents and around 28 publications in international and national peer-reviewed journals. And sir is a life member of Laboratory Animal Scientists Association, Indian Pharmaceutical Association, and Indian Pharmacy Council, and is also a CPS CA nominee for many institutes. And sir is having around 16 years of experience in teaching and research. We are very glad to have you here, sir. Thank you. And the session is over to you, sir.
Thank you, Dr. Estivia, madam. And sir, can we go for the presentation? Sir Niranjan, sir, with your permission.
Yes, madam. Yeah, Dr. Sucharita, madam. Yeah. Welcome, sir. Dr. Ronald Darwin, sir.
Thank you.
Nice to see you.
Thank you, sir. Thank you, sir. Thank you.
Yeah.
And my screen is visible?
Yes,
yeah,
sir.
visible,
yes,
sir.
Okay. So, 1 minute. So, good afternoon, everyone. Hope you are doing a hectic job, doing continuous presentations, continuous conferences. Congratulations to the principal and the team who is doing a wonderful job. Occasionally, we are getting to know about Seven Hills College of Pharmacy. And today, I take the responsibility of talking to you regarding a few things that is something related to the drug discovery. Something which is closely we are moving towards. Actually, drug discovery is not still an older trend. It is actually moving towards a new trend. And something that has captured the total world has even captured the drug discovery also. Artificial intelligence, what I mean is like the thing which has captured the total world is artificial intelligence. That has occupied even the drug discovery also. Here and there, it has put its footprints. And recently, I was watching some news channel, and finally they said that the news reader, the proprietor, was robo, which is more beautiful, doing the things, pronouncing the language very promptly, and artificial intelligence have actually occupied our day-to-day life. So the thing is, myself as Dr. Ronald Darwin, working as professor and head of the department. This is our Wales University Vistas campus. Only 2 blocks I have shown. One is a pharmacy block and one is the admin block. And so what is artificial intelligence? This is a field which is actually related to computer science. And that is something which has occupied the total world. That is what I was saying in the previous slide itself. A field, artificial intelligence is a field that is actually related to computer science and which is highly involved in problem solving, right? In case of drug discovery, the identification of the lead molecule as well as the proper fitting of the lead molecule into the receptor is a major problem. That has been solved, and that can be resolved by the role or the introduction of this artificial intelligence into drug discovery. So the artificial intelligence can be categorized into 2 things. One is machine learning, and the second thing is what is it, deep learning. So relating to the drug discovery, relating to new drug molecule development, you can say how the artificial intelligence is going to work on that particular concept is like simulation labs. What we have, is what is it, simulation in case of what is it, ocean technology. We will be sitting in a room, but we will be visualizing something that we are moving into an ocean or something. We will be sitting in the, we'll be using a computer or display screen. By that, we will be checking the drug on an animal. That is what you call a simulation. The same thing is applicable here. Simulation of human intelligence, utilizing computer system and other related technology. So what is drug discovery? Even we need to remember that this is a base of drug discovery. What is drug discovery? And how are drugs discovered and needed? Drug discovery starts when there is an emergence of a disease. The need of a drug is a need of the time or the need of curing a emerged disorder. See, for example, COVID was the biggest task in front of us the previous years, starting from 2019. So we choose a disease, or the pharma companies or a research lab will be choosing a disease. And in that particular disease, they will be identifying what will be the target segments, a chromosome, a receptor, just a cell membrane or an enzyme. That will be the target for a particular drug. So we have a problem. For that problem, we are finding a exact solution by identifying the targets. Solution, what is it? Problem is there as well as the target is ready. And we need to identify how to fix this disease with that, what is a target that is actually done by the utilization of a bioassay. Third thing is bioassay. Bioassay that is actually used to determine the biological activity, whether the disease can be controlled by utilizing the particular protocol, but using the particular procedure or not. And after that, find a lead compound. See, you have a disease, you have a target region, you start identifying a lead molecule. That is the thing, that scenario that has been followed generally. Lead compound will be identified, and the structure of that particular lead compound will be identified. If the structure is not known, if at all the molecule that you are planning to develop is an anonymous molecule, right? So the anonymous molecule, if it is there, for example, if it is from a plant source, or if it is from an animal source, if it is from a microbial source, the lead molecule, the structure of the lead molecule is not known to you. In such a case, you determine the structure of the lead molecule. That's the general thing that happens. And once you determine the structure, you confine the synthesis of that particular lead molecule. You do some chemical reactions, you add some substitutions, or you break some substitution that is already existing. You build up the molecule and you produce some analogs. That is what you call the synthesizing analogs of the lead molecule. So after the generation of this, lead molecule, you identify the structural activity relationship. That is actually done by the involvement of a particular group, involvement of a particular substitution. What will be the impact on the disease? That is what you can call the structural activity relationship. After doing that, you synthesize the analogs in a surplus manner. And sorry, just repeat it. After that, after the lead molecule is ready, what you do is you will be doing the toxicity testing in the animal model. That is the primary pharmacology role. You will be testing the lead molecule in an animal model. There are various animal models with respect to this toxicology testing or toxicity testing. There are many guidelines like OECD guidelines, ICH guidelines, FDA guidelines. All these things are the guidelines that are, that has to be followed when you're doing a toxicity testing. Primary toxicity testing will be done by OECD guidelines. So in this case, you will be determining the toxicity of the particular lead molecule, and also you'll be determining the efficacy of the lead molecule. Once you do all these things, you will be going ahead with respect to the pharmacodynamics as well as pharmacokinetics of the drug molecule. This is a basic scenario. And after the thing is over, you say that the chemical or the xenobiotic is an effective agent with a good therapeutic potency. Am I audible?
Yeah, you are audible, sir.
You are audible, sir.
Okay. Once the xenobiotic or the chemical substance is identified to be as potent, in such a cases you will be going ahead with respect to filing a patent for a particular drug, stating that you are the first person to reveal the drug. You have done the preclinical evaluations, and you have identified the efficacy as well as you have identified the safety aspect of the drug. And after following all these guidelines, you'll be going ahead with respect to the filing of patent. So after this, you'll be doing with respect to you'll be studying the drug metabolism, continuing the testing of drugs, and after all this process, you'll be doing the clinical trials. And after the clinical trial process has been done, you'll be going ahead with respect to the marketing of the drugs. And this is a routine scenario. So revolutionizing the drug discovery with artificial intelligence. What is the transformation that you expect from the current or the routine synthesis of a molecule to its development and to its availability in the markets? This is the thing that I've been discussing. A general routine process of drug discovery, and what is the revolution that has been made into the process of drug discovery by this artificial intelligence. You know that one thing, penicillin, chloral hydrate, and warfarin. These are the agents that are being used commonly, very commonly, but you know the history behind them. These agents are being accidental discovery. Penicillin, chloral hydrate, as well as warfarin, and even you can say the sildenafil citrate. Many things are there. And one more thing you can say is like quinidine. Actually, it is quinine has been used, and rather than after that, it has been modified. Structural change has been done, and it has been identified to have antiarrhythmic property. So most of the drug discovery that happened in the history are all accidental, right? So that is a scenario with respect to the 20th century. And these accidental discovery are very useful. They are leading the medical society. These are all the older scenario. So what is a resolution, or what is a revolution that artificial intelligence can bring in the current scenarios? Sophisticated development can be done by this particular artificial intelligence. So one more important concept that I can tell you is like when a pharma company is going ahead with respect to the production of, or when they are starting an identification of a lead molecule, or simply to state, you can say that when they start a new drug discovery, they spend billions and billions of dollars. So they invest dollars expected result is plus or minus. They still spend billions of dollars to take the drug molecule to the market, to hit the drug molecule to the market. So what will be the fate is like they say in a scenario that 89% of the expected drug is killed even in the market or in the lab itself, because either they fail by two scenarios. The developed drug may be toxic, or the developed drug may be therapeutically ineffective. Right? That is a reason 89% of the drug that is being developed as a new drug fails in most of the scenario. And 11% of the drugs will be reaching the market. Even in the 11%, they say that 10% is being rejected. 10% of the drugs has been rejected because of reported pharmacovigilance effects, like the reported side effects, reported late, adverse drug reactions reporting. All these things have made the molecules to be withdrawn from the market. This is a scenario. So, so this scenario, even if it is existing in the current scenario, that will lead to economic infeasibility for further development. So once a pharma company invests this much $1 billion on a particular molecule, there will be an economical or economic infeasibility for the further development of a new molecule. So, and one more thing is the time and cost of investment is so high. They have to pay for employees, machines. They have to invest on the machines, and all these things are too high. And that is a problem with respect to the conventional method of drug discovery. And probability of success is also very low. You can say that the probability of success is also very low. So it is time we can turn around towards a drug discovery by using artificial intelligence. So here, this is a thing with respect to drug discovery. First thing is lead molecule identification. You can see in the pictorial representation. First thing is lead molecule identification. You will be having a disorder, right? I can explain it pretty clearly. You have the disease. Probably you say that there is a new type of emerged, CNS disorder, for example, CNS disorder. You have a lead molecule, you propose a lead molecule, that is L that has been mentioned. I can use slide. Yes, this is a lead molecule, L-ball. This is a type of molecular compound. And this molecule can be proposed or propelled towards a, target, right? Rather than going ahead with respect to the screening of all these multiple balls, you can target only on that particular ball. L is targeted. It is sent to the target, that is the T ball. In the sense, target in the sense, it is a specific protein. It is a specific, it may be a specific protein. It may be a specific genetic sequence. And the binding of this particular lead ball L towards a T ball will cause a structural change. And because of this, binding of this L to T, that will generate an action on the biomarker. That is what I was saying, like structural change, and that will lead to generation of a biomarker. That is called as a T ball. That is mentioned in the last thing, T ball. That will be a measurable indicator. T ball is a biomarker, that is a measurable indicator. And what will happen is, like, once you do this process, it is very clear that you will be targeting only one lead molecule. You will be having a single lead molecule. You will be doing a targeting towards a single target, and you will be having a output, that is a single biomarker. See, all these things, by seeing all these things, it is easy, right? So visually, if I could say, the sequence is very clear. As like conventional method, there is a lead molecule, there is a target, and there is a biomarker, and P is a pathway, how it is moving. That is, everything is there. But these things will be analyzed, and it will be done by a computer-based, intelligence-based, artificial intelligence-based system. So lead molecule, from where this lead molecule will arise? The lead molecule may be a molecule that you have in your hand, or the lead molecule may be a molecule that you are proposed to state, like a plant source, alkaloid, or a terpene, or a phenol, or whatever it is. You say that this is a lead molecule, and you determine the structure of the lead molecule, right? You take the structure of the lead molecule, and you take the lead molecule, feed to the system. Once it is being fed to the system, that will act on the target. That will be binding to the target, and specifically, you will be having different number of proteins, different number of genetic sequences. All these things will be there in a specific nature. Only to that particular target it has to bind. See, from where these targets are. See, these targets are obtained from the patient history. These targets will be taken from the patient. See, there are three sources of targets, basically. One is from the clinical side, maybe from the hospital side. Second one is from the pharma company side. And the third one is from the institutional side. These are the mandatory three sources of this, target. So you have the target. As a researcher, you will be having the lead molecule. You will be procuring the target molecule from three sources, and you will be doing that. So that is a programmed scenario, right? This is a programmed scenario. Once it binds, see, actually, for example, I can say with an example. See, in case this is T is an enzyme. T is a particular specific enzyme, right? Only a specific enzyme. The drug is binding, or the lead molecule is binding to that particular enzyme. What will be the structural change in that particular enzyme? That will happen after the binding of lead molecule to the targets. When a structural binding is being done, that structural change will be identified by this, biomarker. Third concept is biomarker. Biomarker will give a report of what has happened with respect to that particular data, and you will be getting a positive or a negative or a moderate result. These are the three possible results that can be obtained in a person. So when you get this possible result, you can predict the mechanism of action, how it is working. See, movement, how it is moving, where it is binding, what are the structural change that has happened. All these things can be tracked down by movement trajectory. So this is with respect to the artificial intelligence. See, in a single nutshell, if I want to say that you have a drug molecule, you have a target molecule, as like that of the older scenario. This is the target is actually obtained from, what is it, the patient data bank. That is a laboratory report that has been obtained from, what is it, a pharma company or a clinical site or whatever it is. So you have that, you have a biomarker, you identify the mechanism of action with respect to this particular lead molecule. So the problem why we want to discover, go ahead with respect is once again discovered here, the time consumption, all these things. Artificial intelligence, why we need to talk about this at this particular juncture? Because even we are not exposed to this artificial intelligence onto 100% with respect to the pharma companies, or with respect to a pharma researcher, or with respect to academic person. Artificial intelligence, I didn't say that will be the panacea, might be the panacea of this year, because it cut down the high cost, right? It cut down the inefficient process, and artificial intelligence will be cutting down the long development time. Artificial intelligence will be cutting down the unavailability of the right models. So artificial intelligence will be, artificial intelligence might be, what is it, simulation driven drug discovery that will be beneficial. Everyone will be having in their mind that how come it is possible? How come this can happen? How a computer can run these experiments? Is it like the software what we use? Is it like an XFARM software or something? We have a model, we predetermined drug will be there. We click on that, we add to that particular animal or a particular human being, and it shows some graphical representation. It is not like that, right? It is related to that, but it is independent and it is individual, and that will be giving, what is it, real results. Like other artificial intelligence model, drug discovery simulation requires a robust real world patient. This is you can stress out the word real world patients. What do you mean by real world patient? I may be a person who's suffering from amnesia, Alzheimer's disorder. And in such case, my, brain acetylcholine level and the source of my brain damage that has led to Alzheimer's may be entirely different from a person or a friend who's sitting in front. So how artificial intelligence will work is that will work purely based upon world patient data, right? And it will take multiple data from different set of patients, and that will be fed to the artificial intelligence lead software, and that will be, bringing out different programs that is called as algorithms. That will be coming out with different formulas, and based upon these formulas, that will, give a set of results. So see, I was talking about real world patient data set, right? So there are three things that you can, say with respect to the real world, patient data. First one is, I hope I have said in the previous slide, including clinical notes. Clinical notes are something that has been written by a doctor after referring to the clinical condition of the patient. So diagnosis report, all these things will be fed to the, algorithm. That is a formula on which all these things will be fed. Like clinical notes will be fed, like imaging, pathology, biopsy, genomics, proteomics reports, everything will be fed to the computer, right? Third important thing is, response reports. And the patient might not be blank enough, right? He may be, or she may be having some treatment. He may be undergoing some certain types of treatment. In such a cases, what is the response of that particular disorder to a particular drug? For example, once again, I can say like a neural disorder, right? Neuroprotective agent has been given, right? In such a cases, when it has been given, what is the effects, right? What is the response? all these things will be fed in an algorithm. All these things will be fed in a formula. So once this, longitudinal patient data is ready, drug screening process is ready. Right? Once this process, these are the primary core of my presentation. Like, first thing is, a clinical note is very important. A patient may be like, example, like a patient is distressed, a patient is restless. All these things has to be fed with the things. A patient doesn't have a memory. The patient has atrophy. The patient, whatever it is, everything has to be fed to the, algorithm. And reports has to be fed to the, algorithm. And as well as, apart from this, you are supposed to feed about the responses to the treatment, also to the algorithm. So once this is ready, the research is ready. Once this is ready, drug discovery is ready. And one more important thing is there, I was talking about clinical notes, clinical data. Clinical data is sensitive, right? We have a rules, like in the forensic, in case of drug legislation, you might have studied, that is, patient data has to be kept safe, and all this information, or all the information related to your personal data related to your patient is sensitive informations, and it should not be shared with anyone. And these can be done by getting a consent from the patient, or this can be this can be taken from getting a consent from the clinical data side, or the diagnosis report feeding agencies, right? So once again, one more thing is, you say that I have received the data, I have fed to the algorithm, and you say that everything is done, everything is fine. It's not like that. And you are supposed to provide a secure network for this purpose, such a way that the data will not flow out from your area to somewhere which is not a right place. So that has to be maintained properly. So there are a few things that I can add upon. There's a software called as RAS as well as SAS report software. This is actually a software which will be having two subcategories. That is the novel diagnostic and the novel digital data generation companies. They are doing that. What is it? Novel diagnosis and novel digital data. See, we are still, as a human being, we are doing a novel diagnosis, but novel digital data generation is nowhere there with respect to our diagnosis lab. And they are a company, they are providing this. This is a starting point of computational drug discovery, generation and digitalization of a real world patient. So you have a thing, and the data will be converted to novel digital data. The patient data will be converted to a novel digital patient data. And second thing is DAS, data as a service. Companies in the category provides a secure network to enable sharing sensitive real world patient data from data sources. If someone is very much interested, they can have a hints of this particular thing. Data from data sources, hospital, research institute, and some other cases, they can go ahead with respect to the pharma companies who are doing that. That is the second thing. So real world data. What is real world data? That is the public, just human atlas. Everything has to be fed, right? Because the scenario is like a drug is fed to a human being, and that is being evaluated. So human atlas has to be fed. And apart from this, apart from the content that is present in the slide, you can add data from real world data from WHO, real world data from international publishing houses. All these things can also be implemented in this particular thing. Okay, potential role. What is the potential role of artificial intelligence in drug discovery? There are few ways by which artificial intelligence will help in drug discovery. So first thing is target identification, and second thing is molecular simulation, and third thing is prediction of drug properties. These are the three important things. And fourth one is de novo drug designing. And the fifth one is candidate drug prioritization, synthesis pathway generation. All these things will be done by, the concept that is called as artificial intelligence. Target identification, we are very much familiar with that. I hope it is not needed for me to take much of the time in explaining about this target identification, because we have many softwares, even molecular docking persons are doing this target identification, 3D structure of targets, and accelerate the design of the necessary drug. And molecular simulation, it is like generating a molecule, and you simulate the molecule. Simulate in the sense how it moves, the absorption, distribution, metabolism, all the process will be done by this molecular simulation. So simulation in the sense, like what a molecule, when it is entering into the body till it is getting out of the body, what it does, everything will be mimicked, everything will be done by this molecular simulation. Prediction of drug properties, everything will be predicted by artificial intelligence, like toxicity, bioactivity, everything will be predicted by this, drug toxicity. De novo drug design, creating completely new drug molecule or entities unknown to the scientists. That is even done by the molecular docking. See, very, very important thing, de novo drug design. See, we say that in case of pain management, still many synthetic molecules have reached the market. We still believe that morphine is a more potent analgesic, right? Most potent analgesic. It has a phenanthrine, ring structure or whatever it is. It has a ring structure. We give the ring structure. And what will be the condition is de novo synthesis? What will happen is new ring structure or new, anthraquinone or some other compound even can be generated that will be very much suiting that particular target, can be done or generated by this artificial intelligence. You give a drug molecule as your own choice because you like that. You expect that this molecule may have a potent, therapeutic potency, potent effect. And you substitute that. But even after substituting that, you will you have not found that as a close drug molecule that you are expecting. But once what will happen by this process is artificial intelligence will generate a new drug molecule which is unknown to anyone. And one more thing is identification or generation of a new drug molecule is very simple. And the important thing what it does is candidate drug prioritization. The thing is, it will be, giving you structural option. What form, cis form, trans form, what will be the best option of the compound to, bind onto that particular target. That will also be done by artificial intelligence. And moreover, the synthetic pathway generation. See, we say that when you go ahead with respect to computer-assisted drug designing, computer-assisted drug discovery, you will be getting millions of molecules. Right? You filter and bring up with 10 molecules. And among that, you filter and bring out with one molecule. You do a synthetic procedure and you try to do a synthetic procedure and you end up saying that it is not possible to synthesis. You close your research chapter there. But here it is like artificial intelligence will give you an idea or will give a hypothetical idea whether it is possible. It will give an idea whether the drug molecule is can be synthesized or not. Feasibility. That is what you call it as feasibility. The feasibility of the drug molecule generation will be or can be confirmed by this artificial intelligence. That is the very important things. And we are nearing to the end of the session. Milestones that we have reached, that not we, milestones artificial intelligence have reached. Even I am the learner of this concept, right? Early 2020, companies have announced that, artificial intelligence designed drug molecule to enter the human clinical trials. Scientia announced the first ever artificial intelligence when it is way back 3 years. And we still say that it is near, it is almost 3 years gone. July 2021, an artificial intelligence system by DeepMind called AlphaFold predicted the protein structure for 330,000 proteins, including all. July 2021, the artificial intelligence by DeepMind called AlphaFold predicted the protein structures. Whatever the protein structures that we have in our molecule has been predicted by artificial intelligence. So once a human structure, human genome has been ready, targeting is very easy, right? That is being done by, what is it, 21. So in the year 2022, February 2022, in silico medicine reported the start of phase 1 clinical. See, this process is totally started in silico, right? In silico in the sense computer-based design of a drug that has even reached 2022 February. It has reached to the level of, what is it, clinical trial, the phase 1 of the clinical trials. And what I can, what is it, thing in my mind is this molecule is going to be a success because it has passed through a narrow way, right? If it has reached the phase 1 of clinical trials, it is going to clear it up easily because already the data is very clear regarding this particular drug molecule. And in January 2023, a company became the first entity to create and validate de novo antibodies in silico. Antibodies has been generated using, what is it, artificial intelligence. 2023 February, the FDA has granted its first orphan drug designation to a drug discovered and designed using artificial intelligence. So FDA has started approving in silico medicine plans to begin a global phase 2 trial for the drug in this year. So everything is possible, and everyone will be having another side. We will always be a positive person on one side. We too will be having a negative side on the other side, right? So challenges also will be there. Wherever we have what is that positive move, we also will be having a negative energy that will be driving us back. So challenges are there even to artificial intelligence also. And first thing is lack of data. And one important thing I'd like to state from this lack of data is the process is still on, and this lack of data can be beaten up. The thing is, the 330,000 protein structure, genome structure has been, categorized and classified by a single company itself. That has been done by a single company itself. So the lack of data is not a major issue, but even it is a challenge on the present scenario. Till date, or up to this mark, it is a challenge. Artificial intelligence, lack of data. Two ways. You can say that the disease-related data challenges there, as well as the protein-related, the target-related, or the human, related data. There is a challenge. Lack of data, challenge is there. And second concern is ethical considerations, right? Ethical consideration. An artificial discovery can lead to thoughts about fairness and bias, right? In the sense, a drug which is expected to reach to the market can be pushed into the market by this artificial intelligence, because a company which is planning for a bias can do it very simply by using artificial intelligence, right? If the data used to train an artificial intelligence is biased, then the inference will be inaccurate. It will be a worst thing. If a data has been biased, the end product of a drug that you will get into the market will be a worst thing, and that will be a tragedy to the human being. Lack of interpretability. Difficult to explain how an artificial model makes a certain prediction as more layers of complexity are added. See, this is not a simple protocol. It is a complex, because when we say about COVID, we say like we even targeted a single protein, but this is artificial intelligence is not the role with respect to a single protein or a single model. That a drug that has to reach a target has to bypass multiple barriers. You generate a drug molecule which is actually fitting superbly into that particular target. But what are the barriers that it has to cross and reach that particular interpretation is a tough task with respect to artificial intelligence. Although the cost of drug discovery can be reduced, it's still expensive to invest in artificial intelligence because in human being, the disease model or the generation of a new disease model requires new algorithms. New algorithms requires new programmers. New programmers request new inputs from various clinical data, scan reports. All these things will once again, push you into a cost concern. See, the thing can be overlooked. Like if this, a set of data has been maintained, a set of data has been generated, this challenge can be overlooked. Till then, it is also a concern with respect to generation of drug by using artificial intelligence, right? So this is with respect to the comparison with respect to artificial intelligence and conventional method. Conventional is target driven. You say that mu, kappa, delta receptor, alpha receptor, beta receptor, target driven. And artificial intelligence, yes, data driven. Conventional has their own points of negativity, and artificial has their own points of positivity. And either the way, it is a double-edged sword. But once you want it or not, this is going to be your choice. Thank you so much for listening to me. And I'll be happy to answer if some questions are there.
Sir, thank you. Thank you so much for your wonderful session, sir. And you have given a very good insights about the current trends in the AI and ML as well, sir. And delegates, if you have any queries, you can interact with today's resource person.
First, let me appreciate you. This is once again Neeraja.
Hi, ma'am.
Excellent presentation, but you have given the pros and cons also. It's actually very thought-provoking. And in the area of drug discovery, you said that clinical data is very important now. So I feel in our country it is still in the primitive stage. Is it? Am I right, sir? If I say that?
No, it is not. There was a.
Am I right?
Clinical data, madam.
Clinical data collection.
Okay, it's a tough.
Clinical data collection.
No. Okay. Actually, we have a diverse set of population, madam. In India, the clinical data collection is comparatively easy when you compare with that of other countries. In India, we have a lot of restrictions for preclinical trials. If you go ahead with respect to the guidelines, there are a lot of restrictions with respect to preclinical trials. But clinical trials, many companies are doing, and it is a part of treatment.
Okay,
so that I don't think that there will be a barrier. There won't be a
is it possible
problem with respect to this data collections, like patient-related scan reports and all these things?
Hello, am I audible? Hello.
You're audible.
Yeah, and one more thing is regarding this data collection from the institute side. Can we go and have the data, and can we use that for the drug discovery? Suppose if I want to test a molecule for the diabetes, is it possible now in India go and collect the data from the hospitals and use that for this one analysis? As it is new to me, just I wanted to know if it is possible. We'll think in that direction.
Okay. There was a technical issue. My line got disconnected. Can you please repeat it again, madam? 1 minute.
Neeraja, madam. Neeraja, madam.
Ma'am, am I audible?
Yes, sir.
Yes. Am I audible?
Yes, now
Yeah,
you're
yes,
audible,
madam.
ma'am. Could you repeat the question, please?
But the line is, some interruption is there in the line.
Yeah, is it clear now? Suppose
Yes,
if I want to do some research and I'll go for the, plant collection, and finally I'll get the target molecule also. I wanted to test the molecule before, in the line of drug discovery, anti-inflammatory property. So for this knee pains and all, if I go and have the data from the hospitals directly, from the institutes, is there that provision is there, or it is not allowed? I want to know that. See, for example, you have the CMC. Can I approach them and get the data, clinical data? Hello? Hello?
Madam, you're audible. I think there's some technical issue with the resource person now.
I think, okay, I'll write to, just leave his email. I'll write it. I don't want to kill the time. So I'll write my question to the email. Thank you very much for giving me opportunity to interact. Thanks.
Yeah, thank you so much, ma'am. And so now I request our respected principal, sir, Dr. M. Niranjan Babu, sir, to kindly felicitate today's resource person, Dr. Dorvin, sir. Sir, kindly do the honors, please.
Yeah. You can go ahead, madam. I think.
Yes, sir.
I'm going to give a memento.
Yes, sir. We are sharing.
Yeah.
Yes.
Yeah. Good afternoon, everyone. I'm honored to be here today to felicitate Dr. C. Ronald Darwin, Professor, Head, Wales University, Tamil Nadu. I would like to state and start by congratulating you on your hard work and dedication, sir. As you know, the field of pharmacy is constantly evolving with the help of artificial intelligence in the drug discovery. Heartily congratulations to you, sir. Please accept this memento for token of respect. Thank you, everyone. Have a nice day. Thank you, madam. Over to you.
Thank you, sir. Thank you for your wonderful acknowledgement. And sir, with your permission, can we go for a vote of thanks for the day one, sir? Here is the time for conclusion for the day one, sir. We have completed 3 sessions.
I think, madam, that whatever the videos you want to show now, you can play that one. University, SU University and 7 Hills.
Yes, sir. Now we can do it.
Okay.
Yes, sir. We'll do it, sir.
Welcome to the land of Balaji. Welcome to the land of pharmacy, 7 Hills College of Pharmacy.
institute provides all latest instruments, ambience of world-class green campus, and best teachers and researchers who are working round the clock for the benefit of students. I am proud and fortunate to be a part of this institute, which is fulfilling every aspect for the benefits of the student.
Pharmacy profession play a vital role in the healthcare system. Someone College of Pharmacy is providing good facilities and good education to all students. I am very happy to be a part of Someone College of Pharmacy.
Is a center of excellence in pharmacy education and research, accredited with NAC A grade. College of Pharmacy is always dedicated to work and to be as a global leader in the development of pharmacy education, research and practice.
College of Pharmacy is not just an institute, it's our family.
Madam, Dr. Sucharita, Madam, can you go for again to felicitate Ronald Darwin, sir? Because that time no sir was not there, actually. I think you are in mute, Dr. Sucharita, Madam.
Sir, yes, sir. Yes, sir. Sir, Darwin, sir, thank you so very much for attending this session. And I request our principal, Dr. M. Niranjan Babu, sir, to do the felicitations to today's resource person. Sir, kindly accept this.
Yeah. Once again, good afternoon, everyone. I am honored to be here today to felicitate Dr. C. Ronald Darwin, Professor,
माता पिता की जिद को उनकी सेहत के आड़े ना आने दें।
University,
उन्हें
Tamil
डॉक्टर से
Nadu.
शिंगल्स और उससे बचने के बारे में सलाह लेने के लिए बुलाइए।
Congratulating you
अपनों को शिंगल्स से बचाना संभव
for your hard work and dedication. As you know, the field of pharmacy is constantly evolving.
माता पिता की जिद को उनकी सेहत के आड़े ना आने दें। उन्हें डॉक्टर से शिंगल्स और उससे बचने के बारे में सलाह लेने के लिए बुलाइए।
वैक्सीनेशन द्वारा अपनों को शिंगल्स से बचाना संभव है।
Thank you, sir. Thank you.
Thank you, everyone.
I'm extremely sorry once again. There was a network issue. I could not answer the questions.
It's okay, sir. No problem.
I'm so glad to be on the stage, sharing the stage with eminent persons. And your institutes, I was browsing through the internet. It's a very big institute. You're doing a wonderful job, sir. Once again, I would like to personally thank Dr. Shravan Kumar for making me to be here on this particular occasion. Thank you, everyone. Thank you. Thank you.
Namaste.
Thank you, sir. Namaste. Sir, can we have a video of Sri Venkateshwara University, Tirupati, sir?
Vote of thanks. Yeah,
Sri Venkateshwara University, Tirupati, sir. Yes, sir.
Yeah, SU University video you can play, madam.
Yes, sir. Yes, sir. Thank you, sir. Respected delegates, thank you for your patience watching. And sir, with your permission, can we propose a vote of thanks for day one, sir?
Yes, madam. You can proceed.
Thank you, sir. And I now request Professor Pushpakumari, madam, to propose a vote of thanks for day one of this two-day international workshop on recent advances and developments in transdisciplinary research. Madam, kindly do the needful. Thank you.
Thank you, Dr. Sucharita. A very good afternoon. On behalf of the management and Honorable Principal, Dr. M. Niranjan Babu, sir, Seven Hills College of Pharmacy Autonomous, and the organizing team of the two-day workshop, it's my immense pleasure to propose the vote of thanks for the day one of the two-day international workshop on recent advances and developments in transdisciplinary research, which is jointly organized by Sri Venkateshwara University, Tirupati and Seven Hills College of Pharmacy, Tirupati. It's my privilege to thank Professor K. Rajareddy, sir, Vice Chancellor, Professor O. Muhammad Hussain, sir, Registrar, and Professor C. Apparao, sir, Principal of Sri Venkateshwara University, who have provided valuable time in spite of their busy schedule in order to enlighten the delegates with their knowledge and experiences. And I also take the honor to thank Professor C. Apparao, sir, Professor Suresh Babu Naidu Krishna, sir, and Professor C. Ronald Darbin, sir, for enlightening the knowledge of all the delegates and distinguished participants by their eminent speech in the session 1, 2, and 3 of day one workshop, respectively. I thank you very much, sir. And I also thank all the delegates for patiently listening and sparing their valuable time for this workshop. I thank all the teaching staff and non-teaching staff who have directly and indirectly participated to organize this event successfully. Thank you, one and all.
Thank you so much, madam. And sir, can we attend on the program with Nishma?
Yes.
Thank you so much, sir. Thank you so much.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, one and all. And that's all for